We are pleased to present the research highlights from the Division of Allergy and Immunology. We believe in sharing our ideas, our discoveries, and our advancements. Dupilumab, which blocks the shared IL-13 and IL-4 receptor, is the first drug to receive U.S. FDA approval for the treatment of eosinophilic esophagitis, also known as EoE. This is a victory for science, rare diseases, patients, and advocacy, and is a launching point for developing more treatments for EoE and approved treatments for other eosinophilic gastrointestinal diseases beyond the esophagus. Scientists at Cincinnati Children's Hospital took part in an international clinical trial of dupilumab in adolescents and adults with eosinophilic esophagitis. They found that histology and symptoms improved when dupilumab was given once a week, which is now the FDA-approved dosing regimen of dupilumab for EoE. This trial is the first to demonstrate that dupilumab effectively treats EoE. Reviewing the fundamental mechanisms of EoE pathophysiology, our researchers discussed how allergen-induced type 2 immune activation by the esophageal epithelium propagates a cycle of impaired mucosal barrier integrity and allergic inflammation, eventually leading to tissue remodeling and progressive organ dysfunction. EoE can progress to fibrostenosis. We investigated gene dysregulation in fibrostenotic EoE, its association with clinical parameters and specific pathways, and the functional consequences. Patients with fibrostenotic EoE had decreased endothelial cell T-span 12. We proposed that IL-13 decreases T-span 12 and promotes tissue remodeling through fibroblast endothelial cell crosstalk. Eosinophils are not the only immune cell with increased levels of EoE. Mast cell levels are also increased. We performed a comprehensive molecular and spatial characterization of esophageal mast cells in EoE using single-cell RNA sequencing, and these studies revealed mast cell heterogeneity, local proliferation, and activation that persists even in disease remission. Investigating gene expression of the esophagus at the single-cell level, our researchers found that allergic inflammation blocked the differentiation of epithelial cells, uncovering the dynamic nature of the inflamed human esophageal epithelium and providing a framework to better understand esophageal health and disease. Our researchers identified an eosinophil colitis transcriptome that changes with disease activity and establishes clear molecular differences between eosinophil colitis and other eosinophil GI diseases and inflammatory bowel disease. Notably, unlike other EGID, eosinophil colitis is not related to strong allergic type 2 immunity, suggesting that other mechanisms are at work. Through our sustained commitment to research and clinical excellence for immunological and allergic conditions, we will continue to change the outcomes together. Thank you for your interest, generous research support, and inspiring patient and family participation.